Hi, everyone. My name is Mario Flores, and here we have also Carla Paniagua. And we are from the University of Texas at San Antonio, provided you with this tutorial on the use of spatial transcriptomics to identify the tissue microenvironments. I'm assistant professor, and Carla is my PhD student. She will be graduating this coming semester. So she's at the end of her PhD, and she's been working in this topic. So let's start now. Carla will introduce a little bit about herself, and once she start talking, and let's look at what would be the logistic that we're gonna use to provide this tutorial. So we have this topic of the identification of tissue microenvironments using high resolution spatial transcriptomics. And we're gonna be showing you some slides just to give you the general uh, uh, pipeline, the general blocks of the pipeline. Then we're gonna go through through the Zoom. Yeah, turn on your camera, Carla. And we're gonna go through some of the code to make it easier for you. We will identify each section of the code. Gonna have three scripts. As you know, in Jupyter, each script has a different section. We call it section or number that identifies the block of code. So we're gonna call it script one dash section 1, 2, 3, etc. So that is easier for you to go through the also data and through the scripts, and that we identify where are you in the process. So go to the next one, please, Carla, and let's get started with this tutorial. So as you can read here, we're gonna provide you with a practical experience in the use of a spatial transcriptomics. This is high resolution spatial transcriptomics because we are using nanostream CosMX. We will provide more information about this, but the important thing for now is that is a high resolution, and that means that you can see the transcripts inside the cell. You have the resolution to be able to identify transcripts within each cell. So some cells may have 400 transcripts. Each transcript may belong to a different gene, and that is the idea that after doing a process called segmentation, you have the transcripts for each cell. So we're gonna be covering the some of the processing techniques. Some other they are already processed because they're provided by this technology CosMX. So we will just mention the ones that are processed, and then we're gonna go to the ones that we did. That is the pre-processing techniques like the integration and the use of deep learning algorithms to identify, to characterize the tumor microenvironment. At the end, you should have an introduction. This is really, Carla has been spending years working on this. So this is an introduction that can guide you for provide you some code so that you can continue uh, searching and looking yourself into the different techniques that you need to process this data and identify the tissue microenvironments. Please go to the next. So these are the objectives. We gonna use some methods to pre-process spatial transcriptomics data, apply common deep learning algorithms. We gonna integrate the different types of data into a final data set that we use with deep learning algorithms that are very novel, like the transformer and graph out autoencoder, in order to process this that is we think is a multimodal data, uh, spatial data. We're gonna show in a little bit what we mean by that and what are the different types of data that we have. Then we're gonna use this we want to identify the microenvironments, and we will interpret these these spatial microenvironments and apply or show you some introduction, some ideas of how to apply the downstream analysis, like the differential gene expression between different microenvironments. Good. Yes. So, what is high resolution? It is a technology that provides high resolution spatial transcriptomics. We can quantify about 1,000 RNAs. Also, it provides you the possibility to have 108 proteins. And there are different technologies. One of the main ones is nanostring, called COSMX, as we can see in the screen. And on the right, we see the transcripts, where here we identify for a few cells on that plot some of the transcripts by the different region in the cell. The membrane are the white ones, the blue ones that are the cytoplasm, and then the nucleus in pink or red. So each dot corresponds here to a transcript. And we define this color just based on the estimation of where they are located within the cell, just to give you an idea of that is the resolution. So imagine the cell in the center. That is about the number of transcripts that we have for that cell. The number of dots that we see in the image is the number of transcripts that we have. Next one. Here are some of the links. We will provide this presentation also, so that you can go and find the data. It's public data, this data set. You can download it. You just need to register and you will be able to get. We're going to present, because of the size of the data, just a small sub-region of the complete data set. So let's start. What is the data? As we said, we have multimodal data that consists on the morphological images, and that is what we see on the bottom right image, right? The morphological, they can identify, as we know, the nucleus or the cytoplasm of the membrane. And with the other type of the raw data that we have is the transcripts, the spatial transcripts on the left. That, as I explained, each dot consists of a transcript. And on the top, we have the overlap of these two data sets. So we can see that in terms of the transcription, the way the data look, the raw data, is that we have an ID for the cell, and then we have an ID for the transcript, the coordinates of the transcript. So let's think that in that image on the left bottom, the coordinate 0, 0 corresponds to the bottom left corner of the image. So we see that on the data, on the table, or on the left, we see that we have an X global and Y global coordinate. That corresponds to the complete data set. However, this data came in different, what we call fields of view. There are small pieces, right? They divide the image into smaller pieces. So we have also a coordinate that corresponds to that FOV. Here, the idea is that you see that for every transcript that is on the image, the bottom left image, corresponds for every dot that is a transcript, we will have a coordinates. And because it's a plane, we have an X and a Y coordinate that identifies the position of the transcript. Okay. However, with this data, we can derive, and also the technology, nanostring COSMX, is able to derive some other data. using some processing techniques. One of them, as you can imagine, we have these transcripts in the space, but we don't know what transcripts belong to what cell. So we need to go through a process called segmentation that COSMX already do for you. COSMX provides you with this, do the cell segmentation and identify the cells. So what that means is that now for every transcript, we have what is the cell that harbors that transcript, that hold that transcript. That is the cell identification by using the segmentation technique. Now we have also the identification of the cell coordinates. Imagine that now we have identified the transcripts of a cell. Now we can estimate what is the center of the cell. And this is going to be helpful because then when we align, when we overlap the information of the transcripts for each cell with the for morphological image, we can use the center to estimate the overlap, the best position. Another thing that we can derive from this data is the gene expression, because now we have the transcript for every cell, transcript from different genes, so we can just count how many transcripts we have for each gene and generate a new matrix in which we have as rows the cells, and as columns we will have the 1,000 genes. So we can have an integer. We will have an integer that tells us how many transcripts we have per gene. And another of the provided data, provided data by COSMX, is the cell type. Once we have the cells, their transcripts, and the number counts of the transcripts for each gene, we can go also through a computational process that allows to, using techniques we will show, there are several ways to do the cell typing. Then we can identify the cell type for each cell. That is the other derived data that we use in this pipeline. So I'm gonna let Carla now go through the cell segmentation and more specifics, because we're gonna start also with the coding part after a few more slides. Please, Carla.
Thank you, Dr. Flores. Can you hear me? Hello, can you hear me?
Carla, we can hear you perfectly.
Okay, perfect. Thank you. So let's talk a little bit about the cell segmentation. So as Dr. Flores was mentioning to you, we're going to have the images and we have the spatial location of the transcripts. But now the idea is that we need to identify to which cell those transcripts belong to. To do that, what we're going to do is the process called cell segmentation. There are different ways to do this, but specifically the one that is used for this data comes from the paper from CellPose. And what they do is they pass like the images through a neural network, and then they apply some processes where they do masks to try to identify the border of the cells. The result, it's going to be something like this. I don't know if you can properly see that, but after processing the data, you're going to get the border of the cell. So what they do after that is count all the transcripts that are inside that border of the cell, and then they compute the gene expression matrix. Another thing that we obtain from the cell segmentation is the center of the cell. So this CellPose software also helps to calculate the center of the cells, the width, and the height of the cells. So this is the process that NanoString uses. But just for you to know, there are some other options using only digital image processing and not specifically a neural network. At the end, what we're going to get, it's the matrix, the gene expression matrix, where we have all the cells. Here it's cropped because we have so many genes, but we cannot show them all. You're going to have all the cells as rows, and then as columns, we're going to have the different genes, and you're going to get the number of transcripts for each cell for a specific gene. And then, now that we have the gene expression matrix, another important step to process the data is to obtain the cell types. In this case, NanoString provides the cell types, but I'm just going to give you a little bit more information on how you can get them. So there are two main methods. One of them, it's going to be cluster-based, and the other one, it's going to be a probabilistic method. So for the cluster-based, what we're going to do is first apply clustering to group cells into different clusters, assuming that these cells, like the cells in one group, are going to have a similar gene expression. Then, what we're going to do is we have a list of marker genes already predefined by someone else. And then we're going to check how expressed are those marker genes in the different clusters to try to identify the different cell types. So that's like the cluster-based method. In the probabilistic method, what you usually have is a reference data set. So this reference data set, it's going to have like the mean expression of a big group of cells from different cell types. And then you're going to compare it to the expression of each of your cells and try to estimate using a probabilistic method how similar they are. And then with that, you will assign the cell types. So at the end, we're going to get a cell type for each of the cells in your data. In the case of NanoString, they used a Bayesian probabilistic method to assign the cell types, which is the data that we have right now. Okay, so let's start working on our pipeline. So we split this, as Dr. Forrest mentioned, into 3 scripts, but we're going to go like step by step with you together so we understand how to do this. So first we split like in half the pipeline, like the pre-processing, and then after the pre-processing, we start with artificial intelligence model. So for the first step, it's going to be after we obtain our gene expression matrix. In this case, what we need to do is to apply quality control to remove low quality cells, because those cells can be either dead or it can be doublets. So we need to remove those cells. Usually in spatial data, we are less stringent with our filters because since we have a spatial data, then the next step to be to normalize the data. So the data needs to be normalized so cells can be compared to each other. We apply component analysis to reduce the dimensions. We apply UMAP, uniform medical approximation projection, to be able to spatially look at the cells without having to look at the spatial coordinates, like just to group them, which is going to give us like the cluster, well, the visualization for the clustering that we're going to apply after that. And then cell typing. So in this case, our data already has cell typing. We're just going to assign the cell type that NanoString provided. As a second step, since we already also have the morphology images, and what we want to do, like our plan, is to include as much data as possible to do the best tumor microenvironment segmentation. So what we're going to do is go to our morphology images, go to the center of the cells, since we already have the coordinates, and then we're going to crop individual images for each of the cells, just as in here, you can see. By standing on the center of the cell, and then the size of the image, it's going to be the size of the largest cell. In that case, as we know, cells can have different sizes depending on the cell type, but our idea is that even if it's a little bigger, like the image is a little bigger than the cell, it can still include a little bit of the context information. We're also going to build a ball tree graph that is going to give us an adjacency matrix, which is going to tell us which cells are neighbors or not. We also have our normalized gene expression, and then we have to one-hot encode our cell type composition. So for that, basically, instead of having just one column that is going to tell you like the cell type, we're going to have a number of columns. As number of columns, the number of cell types, and then if the cell belongs to that cell type, you're going to have a 1. If not, the rest of the columns are going to be 0. After that, we're going to use our morphology images that we extracted for the individual cells. And as you know, like we cannot use the image directly, so we need to extract information from these images. So to do that, we're going to go through a ResNet 50 convolutional neural network, and that's going to help us to extract the important features of each of our cell images. So now that we have all this information, we want to include it into our analysis, right? So what we're going to do is we're going to have a cell of interest. In this case, our cell of interest is going to be N, and then we're going to have the neighbors of the cells. We're going to put together the morphology features extracted from the images. Our, in this case, this one is the...
Probably you can explain that later because they cannot see the equation.
Okay, yeah, I'll explain
The general idea of this slide is that you see there is some pre-processing, and where we want to get is that yellow matrix on the bottom right. That yellow matrix is also a matrix that has as rows cells, and columns are contain gene expression for the 1,000 genes that we have. However, the expression now is modified. We call it normalized to include the spatial information. Remember, we want the target here is to identify different tumor and characterize different tumor microenvironments. So we want to get spatial information. What is around this cell? The cells, they are not by themselves. They are including tumor cells. They are surrounded by some cell types, and there are different types of microenvironments. Some may be more severe, for example. So that yellow matrix on the right bottom includes now the spatial information that we extracted from the raw data, but in general is the overlap between the transcriptomics transcription data, spatial transcripts, and the morphological images that are just images. That's the reason we can treat them with a very well-known image algorithm, in this case, ResNet 50. So, Carla, now want to go step by step. And that's when you need to open your Jupyter. We will try, because the data that we provide is just a small section. You will try to run it. Now we will see how much we can do. One of the situations with this, the environment that we use, Jupyter, is that it's very good because it can, we have many functions and everybody sharing. At the same time, for some aspects, it's not very robust. If you have a different library, if you have you download a different date, it may not work. That is the way it is. Well, let's try. Go ahead, Carl.
Okay. So before we start, were you able to install the environment? Or do you have any questions on that part? We can start.
You receive a link where there is just a link with all the different code, the data, and also what else is there? Images, I think.
Yes. So I'll actually paste the link on the chat.
So why don't you do it from the beginning? Let's go, Carla, open the browser and go to the link, and then also copy paste the link on the chat, right? Now open your browser, go to the link, and start from zero, like if you were one of our, you were starting.
Okay, can you see my screen?
Yes.
So first, before starting, please open this file. So the first step is going to be to install Anaconda, which I already have in my computer. I hope you already did that. And then the second step is going to be to install the environment. So for that, you have to go to the link. I'm just pasting it again. And then in installation folder, you're gonna find two files. One of them, it's called tgme2mac, which is if you have like a Mac or Apple device. and then TGME2 for the rest of the devices, Windows, Linux, etc. So you're going to download this file, and then you're going to put it on a folder. Let's do that together.
This process, we are trying to avoid for you to have problems in the different versions of the libraries. So what Carla did is this script that will install the environment with the appropriate library so that you don't have to fight with that. Gives you that big advantage in the case of the use of Python.
So,
However, there are differences when you use Windows and Mac, so we will check on that.
So I just created a folder, I called it tutorial, and I downloaded it there, the file. Okay, so let me know, like please try to follow what I'm doing. If you have questions, just let me know and we
Just put it on the chat because I don't think they have audio.
you can put it on the chat.
If you want to stop, if not, go slow, Carla, go step by step. And as we said, go back in between the presentation so that we can make comments of what's going on and then through the code. You can open now your terminal to start running the file, right, to create the environment.
Yes. So, well, you create a folder in your computer, and then on Anaconda, let me open it so you can see it. You're going to go to PowerShell prompt, and you're going to launch it.
This is the case for Windows, right?
Yeah, this is the case for Windows. For Mac, you should also look something very similar. The Anaconda Navigator environment is very similar.
And for Linux?
For Mac.
And for Linux, you will have to download the file and run it locally.
Okay. After that, like you have the file, and then the next thing you have to do is to go to the location where you save the file, right? So we're going to go there. We're going to click in here, and we're going to do CD, and then paste the location. And then now we're on that folder. After that, what we're going to do is, in case you don't have this, you can copy it and paste it. We're going to update our conda to make it like run a little bit faster. So if you have Windows, you can use this command. If you have Linux, you can use this command. Okay. So just paste it. Oh, sorry, there's something wrong. Just paste it. It's not letting me paste it. I'm going to type it down. So it's going to be conda update and base conda. This will just like update Anaconda and help you run faster a little bit the installation. So we're going to wait for a little bit. Any questions so far? Okay. Okay, so now that we updated it, we're going to go and go conda environment create, and since we are already in the location, what we're going to do is pull that file. That file has all the information of the packages and the versions that we need to use. So what we're going to do is call that file. We're going to do conda environment, and then we're going to create an environment. We're going to have to call that file tgme2. If you are using the Mac, don't forget to add like /Mac, and then name and then I'm going to call it tutorial 2, but you call it as you want. And this step might take a little bit because it's going to install all the packages.
So what is happening here, you're installing the same libraries that Carla used so that they are compatible and you don't have any incompatibility problems. That here, Carla spent some time by providing this. It is a great way to transfer your work, and the person that you're sending this code works without having to do a lot, spending time figuring out what are the problems with the different versions.
Any questions? All good? Okay. Are you with me, or did you already do this before?
I mean, if that is the step that you are working now, however, you already have the environment, so you can go back and explain more in the meantime. Leave it running and go back to the slides. Let's go back and continue a little bit on that.
Okay. Well, do you want me to explain all the steps to install it, or just
Yes,
go ahead?
yes. Go ahead, please.
So, well, after it finished loading this step, we're going to have to activate the environment that we created. So it should now have everything that we need to work on. So we're going to have to activate it. And after that, we're going to start we're going to install IPy kernel. IPy kernel, it's going to help us for Jupyter to recognize all the packages that we just installed. So we have to first install it and then run this line. And here you can give it the name that you want. Remember in here, if you just put tutorial, then just put tutorial. If you put a different name, this name has to match. And in here, you can put whatever you want. After that, you will type down Jupyter Notebook, and then Jupyter Notebook will open, and that's where we are going to start working. So after you open Jupyter Notebook, you're going to have to install PyTorch, because PyTorch installation is going to be different depending on what you like your system, what system do you have. So if you have GPU, it's gonna, a good thing about Python, if you go to that link, is that it will automatically detect like what do you have available in your system. So for example, for myself, I have a GPU, so it's giving me the option to directly like install it with GPU. If you don't have a GPU, you can just pick CPU. And then it also has like a different command depending on what system do you have. So you're gonna have to copy that command and then paste it on Jupyter Notebook. Let me, while it's loading, let me open the environment that I had before. Let's go to tutorial. Okay, so I open Jupyter Notebook. To open a new notebook, you just go to new, and then in here you will see the environments that you installed. So I have tutorial 3, which is already installed. I know this one is still running, but I already installed it. in here. And there you will see a new screen. There you will paste the command from PyTorch installation. So just as one comment, it usually puts a 3. You can just remove it. Then you can run it. You will see I will have it already installed and it will tell me, yeah, requirement already satisfied. But if not, it will install it for you. And then, going back to file, after you install Jupyter Notebook, then you're going to have to install Torch Sparse, Torch Scatter, and Torch Geometric. So we're going to go in a new line with Torch Sparse. And then we can do Torch Scatter. I already have them. That's why I'm getting layer requirement already satisfied. And Torch Geometric. So these packages are going to help us to train our model and to, well, in this case, we're not going to train it. We're going to provide it to do a pre-trained model, but to be able to use the model and extract the embeddings and everything that we need to work on with AI. So we already have, like, we installed all of those. After we install PyTorch, now we need to go back to the folder and we need to download the data and the codes. So we can just download the data and the code. Might take a little bit. Just be patient.
Other of the things that you need to know is that these data sets are big. We have a transcript and we have the images. The images are big, big files that you need to have enough memory to process. Here in this tutorial, as I mentioned before, we got a small, really small region, a small subset of the data sets, and that's what we are providing in the folder. And the intention, we are able to process this during the tutorial, and you will do the same if you have the complete one. So there is no difference in terms of the processing. But here with this small data set, it makes much easier to be able to complete it during our tutorial.
So when we download the data and the code, we're going to put them on the same folder that we created and just extract them to decompress them because they are compressed. And that's it. If we go here, you will see that we have now the code and then the data. I'm going to move because we have two. Let's just move it in here. Let's see if we can. Yeah, better. And also this one. Okay, good. So now we have the code and we have our data. And then we're ready to start. Let me just close all these windows that I opened. Okay. So after we downloaded the code and we downloaded the data, we're good to go. Everyone's fine? Questions or any comments?
Now you can go back to your environment to see if it's completed.
Okay. It's still running.
That means that you don't have, and that's a situation that we have seen also when it's still running and half an hour, what you can do before is to stop and do an update on your on the version. What is the, can you comment on this, Carl? I forgot
Yeah, you have to update your conda environment. Let me go
the
back.
conda. Yes, we have seen that keeps running forever, never finish. So what we did is to update your conda.
with these 3 lines. Depends on what you have. I think this one also works for Windows, but this one won't work for Linux. Okay.
In case you have that problem, you can do. For now, Carla already got the Conda environment working. So why don't you work on that one?
Yes.
Do
Okay.
go through the steps that you mentioned of Conda, activate, and all this so that they can do the same.
So here, after you install the environment, you will have to activate it, install ipykernel, and then generate the tutorial environment from ipykernel, so you will be able to visualize it in the Jupyter Notebook. After that, you will type Jupyter Notebook, and then it will open. Okay? So let's go back. I already have the code and the data, so in code, I can go.
So can you deactivate and activate your environment so that they
Yeah.
see it?
Yeah, I can do that. Let me open the prompt. Let me close this. So to activate the environment, you're gonna have to do conda activate and then the name of your environment. And then to launch Jupyter Notebook, you will do Jupyter Notebook. And then you will see that it opens. You will have to go to the folder that we created. I call it tutorial. You will have to go to code and then open the first one. In here, just make sure you select the environment that we created. So in my case, it's tutorial 3.
Because that environment will contain the appropriate libraries. If you use a different environment, in many places, it may not work. So now you have everything you need. But to get to this point, take some time because of the reasons that we explained. The installation of the environment takes means that you are collecting libraries from different repositories and installing it in your system to have the right versions. Once you get to this point, the tutorial, I mean the processing now gets everything in Jupyter. So let's go back to you see one of the things that you can show is that this is the script number 1. As you can see, the script number 1 on the blocks that of code that we see in the bottom, each block has a number. And that is the script number 1. That is what is S1 dash step number 1. And then we have the script number 1 dash step number 2, so that you can go back between the presentation and the code in Jupyter. That make it easier just to identify. If you go back to the presentation, then we see that the first 3 steps are, and please go ahead, Carl.
Okay, so the first step is going to be to load the packages. Second step is going to be to load the data. And then the third one is going to be to create a spatial object that we're going to be working on. So let's start looking at it. We're going to start loading the packages so you can run it. There are 2 ways to run stuff in Jupyter Notebook. You can either just type the run, or you can do Shift Enter by selecting a cell. Okay, so you can see how packages loaded good. You continue loading packages.
But let's go and explain a little bit, right? The libraries that you are using.
Okay,
So
so
in the number 1, okay.
we have annotated data, ScanPy and SquidPy. These 3 packages are going to help us to create the object that we're going to be working in. These 3 packages are specifically designed to work with single cell and spatial transcriptomics data. And they have all the functions that we need to visualize, to create the object, to save the object, to load the object, etc. Then, in this case, well, we're just loading NumPy, which is going to help us process all our data. Matplotlib, it's going to help us with the plots. And then here it's just to visualize the versions. well, here we loaded n data again. This one is not necessary. And then you're going to do NumPy and pandas. Pandas, what it's going to help us is to visualize everything as a data frame. And we will be able to edit the files like as a table, like as if you were using Excel, you will visualize the data frame. Then NumPy, it's going to help us to process the arrays. OS is for directions. Matplotlib is repeated, I just noticed. And then pathlib is for paths, for like defining paths.
So many libraries they already know, and they're very general, right? In this case, all the data that we are using, we are putting it in annotation data in these objects that include also the observations. The rest of the library, like ScanPy, that is a library to process single cell data, and the SquidPy, that is a library that what? Can you repeat that part, Karen?
Yeah, SquidPy also help us to visualize spatial data.
It's a library to help you visualize the spatial data. The rest, we believe that you already are familiar. We assume like NumPy and Pandas that help you to process these data frames.
Yes. Okay, so after we loaded all these packages, the next step is going to be to load our data. We have to change the path, so keep that in mind. You're going to go to tutorial, well, to the folder. You're going to go to data and just make sure you copy the right path. And then you're going to
So
paste.
within the data, remember that there are two types of raw data. One is the transcripts, and the other one is the morphological images. In this case, Carla is uploading everything into the data frame, right? That everything that is contained within the file. And please explain that part, Carla.
Yes, so we have different files in here. The first one, it's going to be the stitch coordinates, like just the coordinates of the cells. We're going to be loading this one first. This one is going to be the expression matrix. Rows are going to be cells, columns are going to be the genes, just as we saw before. And then this other file, it's going to give us the cell types for each cell, what cell type it corresponds to, which was provided by NanoString. Then mosaic will be the morphology image. So as you can see, we can see all the cells. It has a very good resolution if you zoom in. And then the other long small, we're going to obtain it through the process, but I put it in case you don't have a GPU and your computer is taking a long time to extract the features of the images. So in that case, you can just take this one. But if not, like we're going to obtain it through the process. So let's start. Remember, we need to change the path to where we have the data saved. We're going to load it. And you can see how we have the coordinates, X global, Y global. We also need to load the expression matrix. So we can do that. And you can see how now we have the expression matrix. We have this number of cells, this number of genes, and then you can see it's very sparse. We have a lot of zeros, which is a common thing on spatial transcriptomics and single cell data. And then, well, this is just for visualization. After that, we're going to first create a new spatial object having now the gene expression and the metadata that we just loaded. And here you have to define paths where you want to save the data. So you can just modify it and put the one that we like you have on your computer. For example, this one. And then save the paths also where you want to save it. For example, I want to save it like in a folder results or whatever. These are just paths. You don't need to worry much about this. And here you're going to have 15. The number of spatial domains, but we're not going to be using this parameter yet, so you don't have to worry about that. So now we're just going to rename the expression to counts instead of exp. Then the next step is going to be to extract the spatial coordinates. So now we are going to create a new object, a new data frame, and we're going to call it coordinates, and we're going to save the global coordinates of our cells. Then we're going to convert them to an array because we're going to use them further on to save them in the object and let them know like the spatial location, exactly X and Y coordinates. The
But
next step,
when you look at the coordinates, even that the name is global, in this case, as we said, we have just a small region. So those are the coordinates of the region, right?
yes,
Of the region that we got. You can go
yes,
back also between the slides so that they can see
yeah,
what's going on, right? We
yes,
already covered the first two steps, right? You
yes.
need to
And now
load
we're
the
here.
packages. Even that you already have the libraries, you're still within the script. As you know, in the case of the Python, you load your packages. The second thing is that we load the data. From this data, we have the raw data and we have the data that is derived from the raw data that is provided by NanoString. Those are the first data sets that we are loading. And now, Carla, the next one is that she create the spatial object, right? What do you call
Right.
a spatial?
It's called annotated data or A data for short.
Okay, what you are loading in that, in the step number 3.
Step number 3. Okay, so for step number 3, to create the object, we're going to need a couple of things. The first one, it's going to be the spatial coordinates of the cells, which we are getting from the metadata. Second one, it's going to be the gene expression of the cells. And the third one, it's going to be the morphology image. So those three we need to load, and then we are going to put them together in the spatial object. After that, we're going to be able to start working on the process. So this is like just preparing our data to be in the right format and to put everything together into one object. Okay, so let's go back. After we do this, we need to load the image. So remember always to change your path to where you have the image saved, like the data saved.
The
And you will see,
intention is that Carla now is gonna put this data in a format that she can overlap, right? The morphological image, the output. I mean, she can select the morphological image and extract the features, right?
right,
And then what she called that the spatial object is the matrix that we show in the general uh, pipeline, in which the information of the spatial information together with the expression information will be used to generate a new expression, some modified or normalized expression that includes the spatial information of the cells.
yes. Okay, so we load the image. First we see it gets loaded as an array, but we can also visualize it. And you see now we already have the image there. Here just to change the name of the image because we're gonna use it further. And now we're gonna create now the object that we're gonna be using. So we're gonna go and add the spatial coordinates first because we want to be able to identify specifically where are each of the cells located. So we're going to do that. You will see now the object has this number of cells, this number of genes, and the information of spatial, which is those coordinates that we just added. Then let's go to the slides. So we finished with step number 3. We're going to go with step number 4, which is the normalization of the gene expression matrix. So in order to make cells comparable across each other, we need to normalize them. So for that, we're going to first normalize the gene expression. We already have like the packages already provide us the function, so we can just call it and normalize our data. We're going to find the principal components. We're going to identify neighbors between cells so we can compute the UMAP, uniform manifold approximation and projection. And then we're going to identify clusters.
So in this case, we can think in the spatial transcriptomics. We can, once we do the cell segmentation, we can look at the data like if it was a data set of single cell. In this, a data set of single cell, we know that we have a matrix that can be, we can visualize this as a matrix where the rows are cells 1 to n, and the columns are the count of the transcript for every gene. We have as many columns as genes we have. In this case, in this data set, we have about 1,000 different genes. And we have values for each entry in this matrix. That is an integer that tells us the number of transcripts per gene for each cell. Now, the data as we see it is not, we cannot process it like that. We cannot believe that this matrix will really represent the number of transcripts per cell and that we can compare between different cells because there are still some technical artifacts. So there gotta be a way, there gotta be a normalization so that we can compare, for example, cell number 1 with cell number 100 and see that they have about the same expression, right? The same number of transcripts for the 1,000 genes or not. So that's the reason we need a normalization procedure that now Carla, after going from the spatial the transcriptomics data, now she formatted in a way that now we can look at it as a single cell data set. And now you can continue with the pipeline for this, right? To process and identify, to normalize. And then once it's normalized, we can use this normalized expression between cells in order to identify clusters of cells that have the same expression and that we can name with a cell type.
Yes. So we're going to have the gene expression of the cells, and as Dr. Flores was mentioning, we're going to normalize it. To normalize it, as I already told you before, we already have a function that is going to do that for us. So we're going to run it. That function, what it's going to do is for each gene in one cell, it's going to divide it by the total transcript expression from that cell. And then you're going to multiply by a factor. It can be 1,000, it can be 10,000, whatever factor you decide. It's just to make the values a little bit larger since the matrix is very sparse. And then we're going to apply logarithm to the values to make them comparable. Then we're going to find the principal components, the neighbors of the cells. We're going to compute the UMAP, and we're going to identify the clusters. So after we run all of this, we can now visualize the results, right? So let me go to the slides. After normalization, we're going to get the normalized gene expression. So as you can see, we don't have integers anymore. Now we have these numbers that account from like all the genes in one cell, the division. So for step number 5, we should visualize a spatial object. So we're going to look at the UMAP with the clusters that we just computed with all these steps that we just did. And we are also going to look at the morphology image and then the cells overlap. And we can also visualize these clusters, but in their spatial location. For example, this green one, which is number 2, in this case, is this part in here. Okay, so let's go back to the code. And then we're going to visualize the object. We're going to compute the UMAP, and we're going to compute the scatter plot to visualize. But first, we don't have we have not added yet the morphology image to our data, so we need to add it. So for that, we just need first to calculate the spatial neighbors, then to add the connectivities across cells. So if you can see, like here, it's marking some edges across neighbors. And then finally, we're going to add the morphology image to our data. So in this case, we're telling it to add a like spatial key. It's going to be called spatial, where we're going
You
to say
need
that.
to go slower and explain in more detail the previous ones, I believe.
Okay, so let me go back. In here, what we're just doing is plotting the uniform manifold approximation and projection that we generated to visualize the clusters.
So these
After that,
clusters are shown,
yeah,
they are regenerated using the gene expression, right?
yes, they are generated using the gene expression, and what we assume is that cells with a similar expression are going to be grouped together in the different clusters.
So here we see about 14 clusters, no, or 15 clusters.
15, because it starts with 0, right? Yeah.
And we see that they are, besides being in a specific region, right, in this UMAP of 2 dimensions, we have the UMAP 1 and UMAP number 2. We see that they also give them a color, right? And this color indicates, Carla?
This color, the colors are going to indicate different clusters.
Okay, so as we can see, for example, for the color that is on the left, that is green, that corresponds to cluster number 2. And those can be possibly identified as one specific cell type. All these cells share this a similar gene expression.
Yes.
Okay, and then the next step is to,
Visualize now in the spatial location of the cells. So the clusters in here are going to be the same. This group of cells, we can see that they are spatially located in here, in this region. So basically now, instead of visualizing the UMAP, we're visualizing it in spatial locations.
so we have several cases, right? In the case of the green, we can see that they form a cluster in the UMAP. You go a little up. Now, once you mark these cells as green, we're not given a cell type name yet. Once we have this and we put them a name green because they share the same gene expression, we can go back to the spatial, how they are distributed. And we see that the green cells, in this case, they are close to each other. However, there are some others, like the orange ones. Orange ones form a cluster, which indicates that they may have a similar cell type, but they are distributed everywhere when we look at the spatial information. Right now, we know we gave them a color to each cell. And in this plot that we see here of the spatial transcriptomics, each dot corresponds to a cell, the original cell in the slide of tissue that we obtained for this analysis. In this case, the data set is for lung cancer, right?
Yes.
Non-small cell lung cancer. So
Non-small cell.
since they have some samples from tumors of non-small cell lung cancer, we expect to have that we have, and they provide that as part of the cell types, some cells that are classified as tumor cells. In this here, the green, what is the name of the cells for the green, Carol, that the NanoString provide?
The cells from the green, the cell type,
Yes,
it's tumor.
those are the tumor cells, right?
Yes.
So they did their analysis. They tell us this is the tumor. These are tumor cells. Now we can see that they express similar, and also they have a spatial location. They're close to each other. So the tumoral cells are there. However, we
Yes.
see other types of cells represented as a different color within the tumor. And that's what we're gonna be showing next. However, this information is just using the transcription, right?
Right.
And the analysis that we are doing includes that we can now make it multimodal. We can integrate it with another data set. In this case, the other data set are the morphological images. And we know that also there are changes in the case, for example, of the tumor cells. There are changes in terms of the size and the shape of the cells when we see a tumor. And that information we hope that that we can use in order to increase this the spatial information that we have for these cells.
Let me take a break of 5 minutes to use the restroom, and I'll be back.
Sure.
Just 5 minutes. Thank you.
So let's all take a break because Carla is sharing the screen. If you have a question in the meantime, it's fine. If not, we will see you at 10:11. And your time is 11:11 for those of you that are in the Colombia time. You put a question in the QA. We don't QA. Okay. Is the spatial transcriptomics data and the tissue staining obtained exactly from the same tissue sections? The answer is yes. They are lined and they are from the same, exactly the same sections. That's the reason now we can overlap at the level of single cells, which is an approximation that we do using the center of the cells obtained from the spatial data after segmentation. We estimate the center, and now we cut from that center the cell in the morphological data. As I said, there is some error there. There is an estimation. Any other question, or does that explain answers? No, any requirements for the staining? What is staining? That is a question that is more for wet lab. What I know is that there are no specific requirements. In this case, they use staining for marking the nucleus and the border. The specifics, I will have to check. But however, all these informations about the staining are at the website. There are several white papers or papers. They're not journal papers. It's just information about what they use. All the specifics for the wet lab experiment for the experiments are there. Any other question in the meantime that we wait for Carla? If not, then you can also take a break of about 2-3 minutes more. Okay, Carla, let's continue now. So we are here at we see the spatial information, right? If we go back to, let's go back to the slides and see where we are, right? Because that is the reference that we have for the pipeline. So we are at this step number 5, that it corresponds to the visualization of the object. Now we cluster the data, we visualize the data back spatially, right? And now we need to include the morphological images. For that, we need to do the step number 6 that consists in cropping the cell images. So go ahead, Carla.
Thank you. Okay. So for that, we need to add our spatial images. So before adding our spatial images, there's just like two steps that we need to add. The first them is to identify the spatial neighbors. So basically, it's just gonna look for each of the cells. What cells does it have around? And then here it's just to visualize it. So as you can see, maybe it's a little bit hard to see, but it draws edges between cells, like this cell and all the neighbors that are around it, for example. This is just visualization, and this step helps us to create like to put the morphology images below and the cells on top. So in here, this is where we are going to be adding the image. So we have to add, we have to say where we're going to add it in our a data object. So if I already run this one, but as you can see, it's going to be added in a data.ons, which is like a section of our object under spatial, spatial, here, spatial, and then the library ID that it's going to use, it's going to be tissue. And it's going to be added the images and then the high resolution image, which is the image that we just saw in here. And then you just need to add scale factors. In this case, we're using a scale factor of 1 and then diameter of 3. So after we run this, we should be able to visualize our data now with our morphology image on the back and our cells on the front. So let me make the cells smaller so you can actually see that they are overlapping. As you can see, like I just made the cells smaller, and you can see how we have our spatial image on the back. And
We
on
have the
the top,
morphological images now, right?
yes.
So you can make two. Please open another section in which you make the opposite, right? After this one, now you make the cells larger. And this, we see that this is the overlap of two data sets right there. Transcription and the morphological images are within the same data set now. Okay, it's just to show that. Now you can continue. The next step is?
Okay, so let's go back to the slide. Step number 6 is going to be to prepare the data to be able to crop the cell images. So let's go back there.
So what we showed before is the complete image, right? The overlapping
yes.
The transcription data.
Yes.
Now we're gonna get, for each cell, we're gonna get one image, one morphological image.
One morphological image.
For that, we're gonna approximate, right? What is what we will get. It's an approximation. Okay.
Yes. So here we're just going to define the path where we're going to save the cropped images. So if you remember at the beginning, here we define the path. So this path, it's going to be on results. So we're just going to tell it like here is where we're going to be saving the images. And if the path doesn't exist with this line, it's going to create it. Then we're going to change the name of the coordinates. Like, well, first just keep the coordinates, and we're going to just add a couple more columns to change the name, just because I had the code like that. So it's just changing the name. You don't have to really worry about this, these 3 steps. Then we're going to add the cell types to our object. So we already know what cell types are because NanoString provide them. So you're just going to load them. Or wait, don't forget to add the path before. So make sure you add the path and then select cell types, and then we load the object. In here, we're just selecting the cells because this cell typing is from the entire tissue from NanoString. So we're just selecting the cells that are in our small section. And then we can visualize how we have endothelial, natural killer, endothelial, fibroblast, etc. And
So
in
now
here,
each cell, identified cell, will have a cell type, right? This is what you say.
yes, each identified cell will have a specific cell type.
We
And
add
then,
in everything in the same object, in the same data frame,
exactly.
right? In terms of
So
the programming, in terms of Python.
yes, so we're gonna copy like the cell type into our object, and we're also gonna copy the width and the height of the cell, which were also provided by nanostring and obtained after cell segmentation. So when we do that, if we visualize, now we're gonna have the cell types here in our object, the width and the weight. So whenever you wanna observe like the metadata, you can just do a data.obs because that's where the metadata is gonna be saved. And then now we can visualize in our UMAP representation instead of the clusters, the cell types. So we can see how all this region in here is going to be tumor cells. And then we can identify different cell types. For example, in here, this one is going to be mast cells. Then we still observe that there are very few cells that were not assigned a cell type. We're going to remove those. And then, like with these two steps, we're going to remove them. And then we can also visualize our cell types now in the spatial location instead of the UMAP. So in here, we're just going to tell it to plot the cell types. And now we can see how those cells that before were the cluster number 2 in green became tumor cells, according to nanostring's cell typing. Okay, so now go to our slides. The next step is going to be to obtain the morphological images for each single cell. So first, we're going to extract the single cell image, basically like divide the images into cells, which is going to be the next step. Then each image is going to be passed through a convolutional neural network. In this case, we're using ResNet 50 to extract the image features. So first, the output of this one, it's going to be a vector of 1000 by 24. Since it's too large, we're just going to compute PCA, principal component analysis, to obtain just the 200 top principal components. Okay?
So here we are extracting an approximation of the image, and then we process using this pre-trained, this very, very famous ResNet 50. That what it's doing is to obtain the features, right, for this cell. In this case, we have images. We have the image for each cell. We have one image. It will extract the features. The big assumption is that some of these cell images will be similar, depending, they have the same features in terms of an image, the same spatial distribution of the pixels. And the output will be now, in this case, an output, a vector of these features that is 1024 features for each cell. Okay, go ahead.
Okay. So let's go back to the code. Now we're on step number 8, cropping the images. So for this, we need to load the packages and the functions. So we're just going to run loading the packages. So history features, it's going to be, let me just go back. It's going to be one file that we have in here in our coding. And we're going to be loading some of the functions in here to extract the morphology features and to crop the images. Then we're going to be loading again OS for path, math function, math library, annotated data. We already have these four, but it's in case like you have not run everything before. PLO, it's going to help us with processing images. This one is going to help us for paths. Again, this one is going to help us for processing images, to obtain distances between cells, linear regression, principal component analysis. This is going to help us to get a bar of the progress of how our image cropping and training is going. and this is just helping you to generate random data. And then these all packages are from PyTorch, which are gonna help us to use artificial intelligence models. So first we're gonna just paste or just run our code. So our code is gonna be looking at each individual cell center. It's gonna find the largest cell, and it's gonna crop the image using the size of the largest cell. And then at the end, it's gonna be saving them on, well, it's gonna saving the location of the images on slices path on our object. So later on, we can go and extract the features of that individual image. So let's run the function. And then that is just declaring the function. We need to call it to actually do our cropping. So we are calling it. It starts cropping all the cells images. As you can see, it's running very fast. And that's it. It's done. It cropped all the images. Now, if you go here, we should have, where did it save? Should be results. Let me just double check my path, and I want to show you where it's saved. It should be inside data. Data, and then results. And here you will see all the images, the cell images that are cropped. So now that we crop all the images for individual cells according to their center, we need to extract the morphology features. For that, we're going to tell it that we want to extract the top 200 principal components and use the ResNet the ResNet convolutional neural network. Here we're going to define the function to extract the features. Right now we're going to be using ResNet, but you can use all of these other convolutional neural networks. And then we're going to first modify a little bit the image, like normalize it, apply some blur, invert it, etc., just to augment a little bit the data. And then we're going to be loading each individual image, passing it through the convolutional neural network, and extracting the features. And these features are going to be saved in our object as a new matrix. So let's declare it, and then let's run it. This might take a little bit longer because now we're running a convolutional neural network. If you have a GPU, you will notice that this step runs faster than if you don't have it, but it just takes a couple minutes. So far, do you have any questions, any issues, something that you want to comment?
One question before if you want to always also look at the Q&A, that's when the questions are going to be. Right now we have one that said, can I, for each of the crop images, find out exactly the transcript expressed? Yes,
Yes.
the idea. Go ahead, Kat.
Yes. So basically, you will see that information also in the gene expression matrix. You don't really need to go to the image, but you can do that because you also have as we saw in the beginning in the slides,
there is an approximation, and you need to consider this. The way Carla did it is, once you have the center of the cell after doing the segmentation of in the transcription information using the transcription data set, you can, after segmentation, identify the center of the cell. And from that center, then you can go into the morphological image, locate the same center, the position, and look around that center and how much you did, Carol. How many pixels around the center you use. Not hear you, Carol. I don't know if you are.
Okay, you can hear me now?
Yes.
Okay, good. So, well, I hope we answered your question, but yes, you can.
No, we couldn't hear any answer. I couldn't. So can you repeat the answer?
Okay. Yeah, so yes, you can find out exactly the transcripts that are expressed by either looking at the gene expression or by, well, you can extract like for the individual cell. If you go to the, let me go back here.
The question that I was asking you is how many pixels you go from the center?
How many pixels? It depends on the size of the cell, the largest cell. So,
Okay, what was the criteria that you use to design?
the criteria that we use to crop the image, it's to check on the largest cell on our data set, and then we are going to crop all of the images to be of the same size, and that size is going to be the size of the largest cells, the width and the height of the largest cell.
So Mauricio, there is going to be some error. You see, if Carla uses the biggest cell, we know that our cell is much smaller than the biggest cell, depending on the cell type or related to the cell type. There is some error. However, it's an approximation in which that we are using. There are better ways to do this, but at this moment is what Carla did. Any other question? If not, please go ahead. And as I said in the Q&A, you can see when there is a question, you will see red. Okay.
Okay. So after this, the last step of this file, it's going to be, let me just go to where we were. Oh. The last step is going to be to save our object. So as you can see, this is just an example of the matrix with the image features. I'm going to show you in the data in a bit. And then the last step is going to be to save our file, the file that we just created. So if you want to see the morphology features that we just extracted, you can add a new line and you can do a data.obs m, and then they are going to be saved here. Image feature principal component analysis. You can visualize them. If you want to visualize it as a data frame, you can just add pandas data frame and they will like be in a nice table. So as you can see, we have all of the cells, and then we have 200 columns, which is like the number of PCAs that we defined. So now we're going to save our spatial object. So make sure you go to where you have like your folder. You copy the path. You paste it there. And let's just run it. Okay. So we completed the first part of our analysis. Now we're going to go and open the part 2. So for part 2, the first step is going to be to load the packages and make sure you run this line to avoid conflicts with the libraries. So we notice this this package specifically can cause some conflicts. So make sure you run this line before. Then let's go back to the slide. The first steps are going to be, give me 1 second. I think I'm not showing you the proper slide. Okay, yeah. So the first step is going to be to load our packages. The second step is going to be to define the paths where we're going to save the files and the variables, again, just as we did before. And then we're going to load the data.
So if you go to the diagram, you already complete and got to the point that you have the yellow matrix, right?
No, not yet. We're going to finish
Okay.
after this one. Yeah.
All right, go ahead. Okay.
Yeah, sorry. So from here, what we got, it's still the morphology features. We now in this new step, we're gonna actually work on that to extract that matrix. So we're gonna be loading the packages. All these packages we had it before. We already saw them. Already talked about them. In here, remember you have to change your path. Let me just change this. And then let's paste it here. This is just for the function to run. We're gonna define 200 principal component analysis. These two values we're not actually gonna use them, but still just gonna run it. And then define the principal components, ResNet. This is also not necessary because we already did it in the previous slide, in the previous script. Okay, so now we need to load our data. Make sure you put your correct path. So you load the data. As you can see, we already have the morphology features in our data. The next step it's going to be to generate the the one-hot encoding for our cell types. So we already added before our cell types. For the one-hot encoding, we're going to convert the one column of the information of the cell types into several columns. So as you can see here, for example, in this one. So this cell, it's going to be a tumor cell. So we're going to have a 1 for the column tumor and then 0 for all of the rest of the columns. So that can be done with one line of code, which is going to be pandas get dummies, and it's going to one-hot encode our data. So as you can see, it generates that matrix with the cells and the cell type. So for example, for 1, it's going to be epithelial cell. The next one is going to be neutrophil, etc. And then we're going to just copy all of these columns into the metadata of our object. So as you can see, we have everything that we did before. Cell types with height. This is the paths for the images. And after that, we start seeing the cell typing. And we're also going to add these two columns in our observations so we can continue processing our data. Oh, sorry, oops. And then you can see how we have all our cell types, and then these are just the coordinates, the centers of the cell. So now we're gonna, from augment, import augment data. So we have this file that is called augment in our code. This one. And then we're gonna be using some functions from that file. It's trying to open it in a different, I'm gonna show you from here. Augment. And then we're gonna be using some of the functions on this file to get the augmented matrix, like the normalized matrix. So to do that, well, first we're gonna save all our cell types into a vector. So you can see we are gonna have 18 different cell types, including epithelial, neutrophil, macrophage, etc. And then we're gonna be using this as input in our functions to tell it how many cell types do we have. Then we're gonna input our object. Then we're gonna give some weights to our data because, let me go back to the slides. Okay. So we're going to be doing the TGME normalization. So basically, we're going to have a modified gene expression, which is going to be a function of the spatial location of the cells, the morphology features, the gene expression, and it's also going to account for the cell types. So now we're working to extract this yellow matrix. For that, we're going to have our cell of interest, which in this case, it's going to be called N, it's going to be the cell of interest. The first step, it's going to have a vector that includes the morphology information, the cell type, if that cell and one adjacent cell are neighbors or not, and then the cell type, one hot encoded cell type. And we're gonna look at each of the neighboring cells of that cell of interest, and we're gonna compute the morphology features. Is this cell a neighbor with that cell or not? And then the cell type of that cell. After that, we're going to calculate cosine distance to see how similar they are. So we're going to have a value between 0 and 1. If these two cells are completely different, the value is going to be 0. If these two cells are completely the same or very similar, it's going to be 1. So this is going to give us like a first value. And then we're going to have an expression. So we're again going to have calculate a similarity between, again using cosine similarity, but now between the gene expression of our cell of interest and the gene expression of the neighbor, and we're gonna multiply it by that factor that we got. So now at the end, we're gonna get a weight that is gonna be used to modify the gene expression. So for the cell of interest, we're gonna have the gene expression of that cell of interest plus the summation that it's gonna be accounting for all the neighbors of that cells, multiplied by each of the neighbors, multiplied by that factor. So if the two cells are completely different, we're not going to modify the gene expression because this is going to be a 0. If the two cells are completely the same, we're going to be modifying the gene expression, giving it more weight to the specific genes that are similar between the cells and the neighbors. Finally, we're going to divide it by the total number of neighbors plus 1 because to avoid like having a 0 down here. Okay, so let's go and go back.
Let me just mention here two things.
Yes, go
If
ahead.
you see, and that's what we expressed, this yellow matrix now contains cells by what we call normalized expression. Normalized expression as a function of the spatial location, but also as a function of the spatial shape of the cells. That is the intention. And also of the gene expression and cell type as a function of what is around the cells. With that, we obtain new values for each gene for in each cell, and that new expression, that normalized expression, now we assume contains that the intention is that also includes the spatial information of these for the data sets. And without now we will use this new expression matrix, right? We can look at it as a single cell data set in order to infer the microenvironments in this data set. Here we are using just a small region. You're gonna see the results in a few slides. But also, you think in the complete data set, it's much easier to see those regions that are tumor regions, those regions that are, for example, infiltrated within immune cells, those regions that some people call like hot tumors or cold tumors, to look at the border. That is what we're gonna see in the next slides. That's what we pretend to do. Is the same thing. We can in some way think that this is what the pathologist, right, will do. Takes the data, they look at the slide, and they start defining, okay, these regions are the tumor, and we see a border, and we see a section that is the vessels. That's what the algorithm is doing, but with no information at all of the biology. It's just the data sets that we have. So please go ahead, Carol. Also, let me tell you that in a few minutes I will have to leave for about half an hour. I'll be back. Carla is going to stay. She's been working in this for years, and this is her PhD project. Go ahead, Carla.
Thank you. Just a comment for Mauricio. I think that error you're looking at, it's because of this line. So make sure you run it before, and then you restart your kernel, and you should be able to run it now. Okay, so going back, where were we? Here. So we define the cell types, and then we're going to input everything together. Yeah, I'll paste the line there, don't worry. Sorry about that. Just run it and make sure you restart the kernel. Okay, so after that, I'm doing the cell typing. We get our vector of cell types, and when we're going to run the function that it's going to give us that new matrix, normalized matrix. So after that, you can see it gives us like the information of the nearest neighbors. It's around like the mean value is 182 pixels. If we go to a data, now we have augment data, and we can visualize those like new modified expressions. And you can see it here. So now we are accounting for morphology image, neighbors, cell types, and like, of course, of origin expression. Then in this case, if we visualize all the cell types that we had, just do it properly, cell types, we can see that for two cell types, TREG and TCD8 memory, we have very few cells. So we're going to remove them for now from our analysis to avoid computational problems. So we remove those two cells, and then that's it. This is our final normalized matrix. So we're going to have to save it. So make sure you copy your path and you paste it there. That goes. You want to put it in data. Okay. And then you save it. The next step, it's going to be to generate the VT3 graph efficiency matrix, which we're going to be also directly inputting into our model for AI. I'm going to explain that in a bit. So we're going to build the VT3 graph by looking at the spatial location of cells, and then we define if two cells are neighbors, we're going to have a 1. If not, they're going to have a 0. So for this step, we need to create the graph. So first, we're going to get the graph from our data using our spatial coordinates, and we're going to be using specifically VT3 graph, where we're going to focus on the top 12 neighbors. So we're going to run this step, and then we can observe our graph. So we're going to have the adjacency labels, but basically what we care about is the adjacency matrix that you see in here. We're going to have 1 if the two cells are neighbors, and 0 if they are not considered neighbors. We can also visualize as a pandas data frame. And then we're going to have to save that graph. So let me copy the path. And we can save the graph down here. Okay. So now that we save the graph, we are done with the second script, and we are going to start with the third script. So if you can go and open that script, you select tutorial 3. Remember always to select the environment that we created. In my case, it's tutorial 3, but it depends on the name that you gave it.
So go back to the slide to look at the script number 3 and what is in the schematic, right? In the pipeline.
It's just the normalized gene expression. Okay, so now we have the AI
So
prediction.
start the yellow matrix. Look at it. In the case, as we know, but some of you, I'm sure, that already use deep learning and before. We know that one of the advantages or the ideas in deep learning compared to machine learning is that we don't need to do a lot of data engineering. However, in this case, we did it. We did some pre-processing in which we include different types of data, spatial data, to make this normalize the yellow matrix with normalized expression based on the spatial information. Uh, the idea is that we include this, make this model in order to include all these spatial characteristics of the cells. And now we're gonna go back to use a neural network, a deep learning algorithm, in this case a transformer, together with a graph variational autoencoder, in order to be able to cluster these different groups of cells. Now the cells include the spatial information. So what we aim is to identify regions where there is a specific cell type composition. That's what we're gonna call a niche within the tumor microenvironment. A niche shares a composition of, for example, tumor cells together with a specific, it may be a specific immune cell. And we're gonna let the algorithm learn and identify these different niches. And then we're gonna name each niche based on the cell type composition, as for example, a tumor niche or a tumor or a T cell niche. You will see that in the next slides. What I think just want to mention is that from here we're gonna use the deep learning. This is the algorithm that we designed that Carla has been working, and includes two types. One is a transformer, and the other one is a graph variational autoencoder. That the intention to use this in the pipeline is to be able to look at the context. There is a context that we can find with a transformer, the context for each cell. If we look at one cell and think what is around that cell, then we can use the transformer, or we are using the transformer that is uh, uh, a model, as we know, a deep learning model use a lot of natural language processing, because it's able to look not just at the company of the other cells around, close to the cell of interest, but it can look far away or farther compared to other models. And then we use a graph variational encoder. Because of the nature of the problem, this can be looked also as an algorithm. That's what Carla is going to be describing in the next slides, and then showing you the code of how we did this. Go ahead, Carla.
Thank you. Okay, yes. So as Dr. Flores explained to you, we're going to be using deep learning to extract the or to identify the different niches. So we're going to start with our normalized matrix. The first thing that we're going to do is to pass it through a transformer model. I'm going to explain more details in a bit. This transformer model, it's going to have 3 attention layers, and it's going to be trained to classify the different cell types. But what we care about is the embeddings that the transformer generates, which have what we call the attention scores, which is going to tell us the interaction between the genes. Then we're going to use those embeddings that we extract from the transformer as input to a graph variational autoencoder with the graph that we just generated in the last step. This is going to have 3 convolutional layers, and then we're going to be generating a new set of embeddings that are going to account for the similarities between genes and also spatial location across cells. And these embeddings, we're going to be extracting them. Our graph variational autoencoder is going to be trained to reconstruct these two matrices. But again, what we care about, it's going to be the embeddings. After we get those embeddings, we're going to put them together, the attention scores from the transformer and the embeddings generated from the graph variational autoencoder, and we're going to pass them through clustering. Clustering, it's going to help us to identify the different niches. And then at the end, we're going to have a set of clusters. Each cluster is going to be a niche. And that niche needs to be interpreted. So we need to do, well, what we do is to check the cell type composition for each of the niches and then assign a name to each niche. So let's continue. So the first step on script number 3 is going to be to load the packages. Then we're going to have to load the data and visualize it. Then we're going to have to load the graph, encode the labels, extract the principal component analysis from the normalized data so it's faster and we can use less computational power to process it. And finally, prepare the data to be loaded. So let's do all of these 6 steps. We're going to go and again load the packages, the same packages that we have been working with. And then we're going to modify this direction. Also in here. And then this has to be 16. We continue loading all the packages. The next step is going to be to load our data. So we're going to call our data. This data already has the normalized expression. Where is it? Here, augmented gene data. And we can also visualize it. Then we're going to define parameters, just as we did on the previous script. And we're going to set the plot parameters. So we're just going to tell to use a DPI of 300. And when we save the images of 500, just to make a good definition on our images and to make them a little bit larger. So the first thing that we're going to do is to visualize our cell types in the spatial location, just to take a look at the plot. As you can see, it gets bigger and the definition is good. We can make the cells smaller. Maybe right now they're a little bit big, so we can just change the spot size, which is the cell size. We can just make them smaller. Then we're going to have to load the graph. So again, make sure you modify the path. And we're going to be loading our graph that we defined before. Then let me go back to the slides. We're here. We need to encode the labels because, as you know, our cell types right now are text. But to train a model, we need to use numbers, not text. So we're going to just encode those labels. So now instead of having the names, we're going to have a number for each of the cell types. And in total, we have 15. Well, it starts from 0 because Python always counts from 0, so we actually have 16. Then we have to extract the principal components from our data to make it faster to run, because if we use all the genes, it takes a little bit more. So instead, you just focus on the principal components. So let's go there. And then to that, we have to normalize the total data, extract the logarithm, scale the data, and obtain the principal component. But we're going to tell it to use now the augmented data that we generated. So we have to run this. This might take a little bit, but not too much. Finally, we save that normalized matrix as X. So X is going to have our normalized matrix. Y label, it's going to have our labels for each of the cells, which means the cell types for each of the cells. And then we can just observe what cell types do we have. So 0, it's going to be epithelial, 1 neutrophil, 2 macrophage, etc. And we have to split our data into training and testing. So today we're not going to train the model. We're going to provide you the pre-trained model, so you don't have to spend a lot of time training the model. But we're going to follow the steps. So first, we're going to split our data into training, testing, and validation. So in here, we see that we have 16 different cell types. We want to use 0.3 as test. And from those 0.3, 0.2 are going to be used as validation. And then we use as input X, which is our normalized matrix, the labels, and also the graph that we just generated. The second step is going to be to create a data loader for the normalized gene expression on the graph. So this will help us to load our data in blocks. We're going to be doing batch, so it just separates the data in different batches, accounting for both the graph and the gene expression. So we first define the function where we're going to be like creating tensors, because PyTorch works with tensors. So we're going to create a tensor for training, validation, and testing, and also for our graphs. So, well, here we're just creating first the data loaders, and the graph are going to be on the next step. So this is for the gene expression matrix. So we input the data that we just separated before, X train, Y validation, etc. And then we're going to say that the batch, it's going to have 128 entries. And then we run this. Now we have to do like to generate a graph loader. Now instead for the matrix, for the graph. So we're going to just define a function to generate the loaders. We're going to tell it that we're going to be using batches. And then again, we have to split the graph, mainly the adjacency matrix. But we have to split our graph according to the training and testing that we are using. So we define that function, and then we are actually going to generate our data loaders. So how is it going so far? Good? Any questions? Let me know if I'm going too fast also. So let's talk about the model that we're going to be using. I already gave you like the general explanation on the model, but let's go step by step. So for the transformer model, so the transformer model that we designed, it's going to capture the gene-to-gene interactions within individual cells by focusing on the gene expression profiles. So it's going to employ what we know as attention mechanisms to calculate the importance or relevance of specific genes within a cell, helping to highlight key interactions involved in the microenvironment. It's going to be trained to predict the cell types. So let me explain you a bit. As input, let's say you're going to have one cell as input to a transformer. And first, it's going to go that input to the first attention layer. So let's say this is our first attention layer. You're going to have 980 genes, right? So each gene, it's going to go inside. And then for each gene, we're going to calculate, well, for each cell, we're going to calculate the queries, cues, and vector. So query, it's going to be the gene of interest. In this case, it will be gene 1. Key, it's going to be a weight that it's going to be representing how related the key is to the rest of the genes. So how affected it is or how correlated it is. And then values, it's just going to be a score that it's always going to be the same for the genes, the different genes. So after we define the queries, keys, and values, we're going to do softmax function. And basically, we're going to calculate the attention score. So it's going to be multiplying the queries and the cues, and then it's also going to be multiplying the values. So at the end, for each gene, we're going to get one attention score for each individual cell, which is going to be representing how similar that gene is to the rest of the genes, or how related this gene is going to be to the rest of the genes. So if you think about natural language processing, you can think about words, and then how these words it's like the context, how of the context of this word, it's going to be related to the context of another word. So that's for our transformer part. And then we're going to have this process, it's going to happen for each of the genes and also for each of the cells. So at the end, the output of this one, it's going to be like the output of, for example, the third attention layer. It's going to be a matrix with the attention scores. the length of the matrix, it's going to be the same, like number of cells by number of genes. And then we're going to have 3 attention layers, and then we're going to have 3 heads, because transformers, like each head, it's actually going to be randomly initialized. So as more heads we have, like better we can capture the data attention scores. The model is going to be trained to predict cell types. So the last attention score is going to be passed through a softmax to calculate the different cell types. But we care about the embeddings that are going to be generated after the 3rd attention layer, which is going to be A3 in this case. And then we're going to the graph variational autoencoder, which is going to be integrating the spatial interactions, like these embeddings that we extracted from here, A3. Across cells with the spatial location or spatial neighborhood information with that adjacency matrix that we generated a couple steps ago. This one. So let me go there. So the idea is like, well, the adjacency matrix is going to be built based on the assumptions that cells within the same tumor niche, will not only express similar genes, but also they are going to be located closely in space. So if we think, for example, about tumor cells, we're going to be assuming that those tumor cells are going to be grouped together spatially. So we're going to take those 2 matrices, well, our matrix and our adjacency matrix, and then we're going to pass them through our graph variational autoencoder. So first we're going to pass it through the encoder, that is going to have a graph convolutional layer, a batch normalization layer, and then ReLU activation, and it's going to have 3 graph convolutional layers. Then we're going to do reparameterization, and that's what's going to help us to generate the embeddings that we're interested on. As I told you at the beginning, this graph variational autoencoder, it's going to be trained to reconstruct the input. So basically, we're gonna get like a reconstructed matrix and then a reconstructed adjacency graph. But we are going to be paying attention to the embeddings generated by the encoder. And then these embeddings are going to be passed through to the decoder, where we're going to have a fully connected layer, a ReLU activation layer, and then another fully connected layer, which is going to finally give us like those 2 reconstructed matrices. But what we're going to do is to extract the attention scores from our transformer and the embeddings generated from the graph variational autoencoder, and we're going to concatenate them for the next steps. Okay, so let's go and do this part. So we're going to start declaring the TGME model. Again, we're going to be loading our packages. Now we're going to be loading everything related to PyTorch, which is going to help us to generate our model. So first we're going to import PyTorch, then PyTorch NN, which is neural networks, functional PyTorch, the graph convolutional network, then how to load, like this one is going to help us to load the data, again NumPy, and optimization just to make it faster. I see that some of them are repeated in here. We're also going to import the transformer, so we can declare our transformer. And then these 3 are also repeated in here. Argparse, which is going to help us to pass the arguments. And then some other libraries, for example, sklearn preprocessing, which is going to help us to, like with the one-hot encode and stuff. Pandas, NumPy, sklearn, which is going to help us to calculate the accuracy matrix. The accuracy value, sorry, not matrix. Matplotlib to plot, OS, Copian system for the paths. This one is going to help us to generate random data. Then this one is going to help us to split our data into training and testing. This one is going to help us to save the checkpoint when training the model. Then we're going to do, this one is going to tell us to calculate like the metrics, metrics like accuracy, recall, etc. And also this one. This parameter is going to help us like to basically the graph variational autoencoder. this is also going to help us for both models. Like a model, just important functions PyTorch needs to run. We're going to do batch normalization and sequential data. And then this is going to help us to get like tuples and other data type. And finally, logging in. So the first step is going to be to declare our transformer model. So we're making it from scratch. So we're going to generate the multi-attention heads. Remember, we're going to have 3 heads. And then here you say, like, you're going to declare the number of heads, number of genes, batch sizes, etc. And then for our first attention layer, let me go back to slides. Attention layer, we're going to say that it's going to be calculating, first randomly initializing the weights of the queries, the weights of the queues, the weights of the values with this tree. We're going to randomly initialize it. First, we're going to create them and then randomly initialize them. And then we're going to apply a softmax. Then we're going to be doing the calculation to obtain the attention scores. So first we multiply, we have the queries minus the keys. Then we're going to do the multiplications all the way here till we extract the final like C product, which is going to be just the attention scores. And then this forward function is just going to help us to like put the order where we want to run all of that we declared before. We're going to have a layer normalization and then the residual connection. So that's the part of the transformer model. Don't forget to run it. Then we're going to have the graph variational autoencoder. We're going to declare packages, which are very similar to what we already loaded, actually. And then we're going to be declaring our graph variational autoencoder model. So in here, we're going to tell it to use input, like to obtain information from the user, the inputs that we're going to be using. And then we're going to tell it to have a graph convolutional or a GCN layer, followed by batch normalization and ReLU, just as we saw in the slides. Graph convolutional layer, batch normalization, and ReLU. Then we're going to have convolutional mean and logbar, which are going to be helping us to encode the data, and then reparameterize the data and initialize our data. Then we're going to have, for the encoder, 3 convolutional layers. We're going to extract the mean, the reparameterized layer, and then our decoder, that it's going to be made of one linear layer, ReLU, and then another layer, or fully connected. Finally, forward, you're just going to be calling them to let them know the order that you want them to be. So we run this. And finally, like we have now the transformer model, the graph variational autoencoder, but we need to put them together. So we're going to do that in here. We're going to tell it to put like the transformer model or in order. We're going to have 3 attention heads, 3 attention scores, layers. These 3, let me go back. Here, attention layers, 3 of those. And then we're going to have a linear, the initialization, and then finally down here. Well, this is just going to feed forward to save our output whenever we get the embedding so we can extract them. And when we say forward, we say that we're going to have, for X, we're going to have the GCN embeddings for the graph convolutional autoencoder. And then for the transformer, we're going to be calling the attention core, the multi-attention layer, as layer, again, multi-attention. And at the end, we're going to be applying softmax so we can identify the different cell types. And in here, we're going to tell it the order. Here it's all the parameters that we need to input. We're going to have our transformer model, and then we're going to have, after our transformer model, of GCN model. And we're going to tell it that the input is going to be X, which is going to be 1 cell. And then the output, we're going to be extracting the different embeddings. And we're going to return that. So whenever we run the model, it's going to bring us all this information back. We're going to set the batch size to be 120, 3 number of heads, number of genes, in this case it's 50 because we're doing PCA. A number of features is the same. Well, this parameter is, you don't really need to put it right now, but just leave it there. Dropout, we're going to have 0.7 of dropout. mode 0, which means that we're not running any specific mode or a special mode. And then in here, you can change the activation functions. Right now, we're not using any, but you can either use ReLU, Liquid ReLU, or GeLU. And we're going to have 16 classes. Then we just generate a random seed. We validate. We put the gain that we're going to have, like more parameters for our model to run. You can define the size of the convolutional hidden networks. I decided to be 32 by 16, but you can also modify that. The dropout, and then finally the number of clusters or number of cell types. And for that part, we're going to be using ReLU. Then we run this, and we're going to tell our model, if we have a GPU, use the GPU. If you don't have the GPU, well, then use the CPU. And then we can visualize our model as a whole. So first we're going to have the transformer, which is going to have the 3 multi-attention layers, and then the fully connector layers, and finally a linear layer. And then it's going to go to the graph variational autoencoder, which is going to have the convolutional layer, batch normalization, ReLU, and then another 2 convolutional layers. So after that, we are going to say for the loss function to use LATAM, and then the learning rate to be 0.001 and the weight decay to be 0.01. Let me go back to the slides, not here, sorry. Here. So after we define the model, now we need to prepare our model to extract the embeddings. So as I was telling you before, today we are not going to train the model because it might take a little bit, especially if you don't have a GPU on your computer. So what we're going to do is go to our folder. We should, if you downloaded the code, then it should already be there. If not, just make sure it's there. And you're going to download either model CPU if you don't have a GPU, or model GPU if you do have a GPU. Make sure you either, if you download the zipped ones, make sure you unzip them. If not, you can just download the PT. Okay, so that file, it's going to have the weights of the pre-trained model, so you don't have to train it again. So let's start preparing the data to extract the embedding. So the first part is going to be to define the data loaders. In this case, since we want to extract the embeddings of all the cells, we're going to be using the entire data set that we have, like this small one that we generated for you to be able to run it. Because we already trained the model, so we just want to extract the features. So we're going to declare a data loader, very similar to what we did before, where we're going to have, like, first we just generate a graph loader for the entire graph at the same time. And then now we're going to do an all data loader with the entire data. Then we're going to prepare to extract the embeddings. So concat, it's going to be X. Like our entire gene expression matrix, we're going to convert it to an array, and we're going to generate the data loader so it has all of our data. And we're going to tell it to use a batch size of 128. For the next step, we need to load the pre-trained model. So make sure you double check. If you have GPU, use the one that has GPU. If you don't have GPU, use the one that says CPU. Remember to put the path to where you saved it. And then you're gonna run it, and I'm getting an error because it says, oh, it's in code, not in data, Codes. Okay. You see how you should get at the end all keys matched successfully, which means the pre-trained model was properly loaded to our system. And we're just going to call it pre-trained model instead of just model. Then we're going to set our model to evaluation mode so we can actually extract the embeddings. Since the model is already trained and you don't need to train it, we're just going to put it on the evaluation mode. Then we're just going to clean cache for CUDA if you have GPU. If you don't have GPU, you can skip this step. And let me go back to the slides now. Well, step number 1, it's going to be to extract the embeddings. So for that, we're gonna be running our model like for the evaluation mode. We're just gonna make sure it's an evaluation mode. We're gonna generate the bar that it's gonna tell us the progress on how it's extracting the embeddings, and then we're gonna load our batch or data loaders that are gonna give us the data in batches of 128. We're gonna pass our data through the pre-trained model, extract the features, and then we're gonna save those features into embeddings. So, well, the embeddings is just gonna be like a new matrix embeddings file now. So when you run this, you're gonna see how it's loading. It's very fast actually, and I'm using the GPU. So if you have the CPU, it might be a little bit slower. So we're gonna save this like the final embeddings into all embeddings. And then we're going to see how we get a matrix of the number of cells that we have by 82. Why 82? Because we have the 50 principal components that are going to be given from, well, the 50 principal components that are passed through the transformer, and then we get the attention layers, the attention scores, and then we have 32 embeddings that are extracted from the graph variational autoencoders. So if you add up these two, you get that 82. And then, since we, like in my case, I'm using the GPU, I need to move my data back to the CPU so we can continue the process. So that's what I'm going to do in this step. Just move it back to the CPU. And then we're going to convert our data, since in this case, remember I told you PyTorch works with tensors, so we need to convert it back to an array so we can visualize it as a data frame. So that's what we're going to be doing here. And at the end, we can see the embeddings. So we have 82 columns, 6,807 cells, which are going to be like all the cells that we had from before. And each of them is going to be having like the set of embeddings. So then we're just going to save this just in case you close it, you want to reload it later, or just to have it. And then let's go to the slides. For the next step, we're going to be doing the clustering. So now that we extracted the embeddings, which are going to give us two main things: the gene-to-gene interactions and the spatial location of the cells. Like it's going to account for these two, and then it's going to be giving us the lower representations. So what we're going to do is to apply clustering after we extract these embeddings, and then we're going to obtain like a cluster. Well, we're going to obtain different clusters for the different cells. So these clusters are going to be grouping cells that have a similar embedding into different groups, which are going to be our niches. Each cluster is going to be representing a niche. So let's start in here. Before I continue, how are you doing? All good? Do you have any questions? Do you have any comments or anything you want to mention? Okay, so I assume no questions for now. So what we're going to be doing next, it's going to be to identify the different clusters. So we're just going to make sure we have pandas and scanpy loaded. We're going to observe our data. So now this data contains all the information that we prepared before. And it also has the embeddings. So the embeddings are going to be saved in, no, we need to add them first to our object. Save the embeddings to the model, to the object. Here it should be object. Save the embeddings in the object. So we're going to save it in the observation part, and we're going to add them. Like the embeddings at the end are going to be in this data frame that we just generated, which is going to be embeddings, and then you can give it a name. Here we gave the deep TME, but you can change the name, like embeddings or whatever you want. So we're going to save them on the object, and then we're going to compute a neighborhood graph with those embeddings so we can visualize them spatially. So we're going to compute it. We're also going to calculate our clusters, and then we're going to visualize those clusters. So as you can see, we generated the clusters. Now we have 13 clusters, 13 different clusters, and we're looking at the different sections. But right now, the different niches. Right now, we don't really know what a niche is, so we're going to have to go ahead and study these clusters a little bit more to get a better idea. But before we do that, let's refine the clusters to avoid mistakes. So basically what we're going to be doing here, this function is going to evaluate each of the cells to check if their clustering assignment is correct or if there are a few mistakes. So for that, the assigned cluster is going to be compared to the assigned cluster of the neighbors around the cell. Let's say, for example, we have a cell and all the neighbors around it are a type, and that specific cell is a different type. It's going to reconsider to see if it reassigns it. So we're going to load the packages, and then here we're going to recheck the clusters. So for that, we're going to compute Leiden, calculate the Leiden algorithm, calculate the adjacency matrix again, and then just refine the clusters to see if we need to modify any cluster. So we're going to run this function a little bit. This is just going to help us to make it a little bit more clear and nice. Okay, it says it's done. Now we're going to visualize the results. So as you can see, we are getting our results a little bit refined. Those dots that we saw in here that were from a different cluster, it reassigns it to the new, to the same cluster that it has around it. Okay. Now that we identified our clusters, we are going to go to interpreting the clusters so we can assign a niche, like a name to the niche. So to that, we're going to be calculating the cell type proportions of each niche. And then the percentage, for that, we're going to be calculating the percentage of the different cell types in a niche, which is going to help us to assign a name to those clusters. So in this part, it takes a little bit of human input. So just for you to to know. So first, we're going to create a new data frame with all the available cell types. And then we're going to be filling it, for each cluster, the percentage that it has for the different cell types, which is going to be done here. So now we're going to be focusing on that deep TME refined domain, like these refined clusters that we identified. We're going to calculate the percentages. In here, it's going to vary a little bit on the version that you have. So just make sure, if you get an error in this part, you just change cell types by count, and then it should work. And then we're going to run the composition. So as you can see, for example, cluster 0 has 52% of neutrophils and then 20% of T cells. So we now need to give it a name, right, to each of the clusters. In total, we have 13 clusters. If you get a little bit more or less clusters than it, just make sure this part matches, because we're going to be assigning a new name to each of the clusters. So in here, I have from cluster 0 till cluster 13. I have 13 clusters, which is good. And then I'm going to be assigning the names down here. So this is where I tell you it takes a little bit of manual input. So for the cluster number 0, we're going to see that we have neutrophils, 52%, and then T cells, 20%, and then a few more of the other immune cells, macrophage, etc. So I'm going to call that cluster immune, because it's made of several immune cells. For the next one, cluster number 1, we see that we have macrophages on 70%. So since most of them are macrophages, we're just going to call that niche or cluster macrophages. For cluster number 3, we observe that we have endothelial cells, and then we also have a few neutrophils, So we're going to assume this one, it's going to be a best cell. So, oh wait, I skipped one, sorry. For cluster number 2, we're going to have 96% of cells that belong to tumor. And if I go back here, I see that this cluster number 2 is this orange part. So I'm just going to call that tumor 1. Then for cluster number 3, now we see a little bit of neutrophils, but most of them are endothelial cells. So I can call this name is wrong. I can call these ones vessels because endothelial cells are the vessels or make the vessels. For the next one, I see that I have 70% fibroblasts. I also have a few endothelial cells and then a very few immune cells. So we're going to call this one stroma. Okay, so we have stroma. That's good. Then for number 5, I can see that I have neutrophils, macrophages, and myeloid dendritic cells. So all of these 3 types of cells are considered myeloid cells. So I'm just going to change it and put it in here. Myeloid cells. Then after that,
Hi, I have a question.
oh, okay, yeah, go ahead.
Uh, in this step, it's necessary to assign one cluster by time and in that order always?
Yes, so you're going to be checking cluster number 0, for example, and then you have to assign the name to that cluster number 0, and they have to be in order. Yes, unless you want to change it like the order in here, you can put 4 after 0, etc. But yeah, it should be in that order. And I see that also we have another question. Did I answer your question?
Yes, thank you. So it's
Okay.
important to maintain the same order because maybe we can assign a different name for our respective cluster, right?
Exactly. So in this case, we get an order, and it's less to make it's less easy to make mistakes, or it's harder to make mistakes. And
I have
is it-
another question. If
yes.
I found that there is a different cluster that there is the same type of cell, can I mix the cluster or join the two cluster in one cluster?
You can do that. It depends. This part, it depends on you. Because since there were identified into different clusters, there might be a few differences between them. So if you want to go in detail and study those clusters individually, I would recommend you to assign them, like, for example, let's say macrophage 1 and macrophage 2, right? It can also
Okay.
be that they are separated because of the spatial distance, like they're just located in 2 different sides of the slide, but they may be very similar between them. So it depends on what you want to do. If you don't really need to go into detail in the macrophage clusters, you don't really have to separate them. You can just put macrophages. So yeah, it depends on your analysis and what you want to do.
Okay. Thank you.
You're welcome. And I also saw that you asked the question of the recommendation of the resolution.
Uh-huh.
In this case, we use 0.8, but you can play a little bit with it. So if you see that you're getting less clusters and you want to have a lot more details, you can just increase that number to get more clusters.
Okay. It,
Yeah, there is
it's
not.
the same like it's the same like in single cell. Like we
Yes,
just put a number to see how is the different scenario for cluster, right?
yes, exactly, because we have unsupervised data, right? So we don't really
Okay.
know like if there's an exact proper value. So we just uh, experiment with it a little bit.
Okay. Thank you.
You're welcome. Mauricio, you got 12 clusters, no 13. That's fine. Don't worry. Just make sure you remove the last line from here, and then you just put 12 inputs in the values. That's fine. So we were on 0, 1, 2, 3, 4, 5. 5. And then for number 6, we see that we have neutrophils. We also have a few macrophages, but since we have 53 neutrophils, let's call this one neutrophils. So this one is going to be neutrophils. Then for cluster number 7, we have 62 B cells. So we can just go directly and call this one B cells. Here. For cluster number 8, we have 91 epithelial cells, 91%. Epithelial cells usually also make the vessels, so let's just put vessels in here. For cluster number 9, we see 97% of mast cells, so let's just call that one mast cells. And then for cluster 10, we have 99% of tumor cells. And we can see cluster 10, it's going to be in here. It's going to be those gray cells that are actually surrounding the cluster number 2, which is also tumor cells. So let's call that one border because it's surrounding the other one. So let's call it tumors trauma border. Whenever you're checking on specifically tumor clusters, I would recommend you to do that. Like check tumor, okay, you see that it's tumor, then you go back and check the cluster where those cells are spatially. So you can identify the tumor border. And then 11, it's also going to be tumor. So 11, it's going to be these green ones, and we can see it's separated from the other one, so we can call that one tumor 2. So let me go here. Tumor 2. And then for 12, we have 96% of plasmablasts. So we can just call it either plasma or plasmablast. Both works. Plasmablasts. And then finally, we're going to go till 13, and we see that's only natural killer cells. So we can call it a natural killer. Like it depends on how specific you want it to be. You, for example, instead of putting natural killer, plasmablast, you can go with lymphoid cells or just call them all immune cells. It depends on how much detail you want to have in your microenvironment. The main idea is that we can identify different tumor regions and the border. So now that I have this and I finished with the names, make sure you run these two, and then we are going to assign those names. And then after we identify the different names, the next step is going to be to visualize our microenvironment. So we're going to run this line where we're going to be just plotting now the microenvironment that we just defined before. Remember, you can change the size of the cells as you want. In this case, I don't want to put the morphology image on the back because like this looks more clear. But if you want, you can put it. And then you just need to remove this part, and it will put the image. And then we're setting a palette, like the colors to plot them, but you can change the colors. Feel free to do that. We're telling not to do like a frame around it. And finally, we're telling it to save it as tgme.png. So let's run this one. And you will see, you will see how now we have this big group of B cells, tumor cells in here. My border is not that clear in this case, but we do have like some border cells. Then we have tumor 2, and we have macrophages around everywhere, stroma, neutrophils, etc. A group of immune cells. So as I was telling you, if you want to be like with like have less details, you can probably divide them like between innate immune system. You can just put immune cells, etc. Like you can have more or less details depending on what you want to, what's your plan. And you can see how we get the vessels in red very nicely. And then we can also count to see how many cells we have from the different cell types. So we see, for example, in here, I have this number of cells for the niche, sorry, for the microenvironments. I have 1,139 cells that belong to the best cells, and then tumor cells, etc. And very important, after we got all of these, let's save our results. Okay, so far, how are you? Do you have questions? Do you have comments? Are you following me? Okay, so if you have questions, just let me know. Finally, that we defined our microenvironment, we can go ahead and do a downstream analysis. Of course, this part is going to completely depend on you and what you want to do. In this case, we're just going to be doing a simple differential gene expression between the tumor clusters, just like to get an idea of the differences between them. So basically, to further study the differences between the tumor-related clusters, we're going to compute the differential gene expression analysis using the Wilcoxon test method. So we're going to be obtaining. Hello?
Yes, I'm here.
Okay, good. We already finished with the defining the microenvironment. We're going to start the downstream analysis.
Okay.
So we're going to obtain the most differentially expressed genes per each tumor niche. And in this case, we're just going to focus on the tumor-related clusters. But you, as I was telling you before, if you want to study any other niche, you can just do that. Like, feel free to do that. So let's start there. We're gonna, in this case, extract the tumor niches because that's what we are interested on. So from our TGME, let's extract the tumor niches. And then you see how now we just have like those cells that were identified as tumor. In here, we're just gonna remove the base because after all the processes that we did, we added this part and it causes some conflict when we wanna rank the genes. So you just need to remove this. Or maybe not anymore because I think that it doesn't have it. So this might work there. So this step might not be necessary anymore. So in here, we're gonna rank genes groups. So let me just tell you specifically what this function is gonna be doing. So we're gonna rank the genes for characterizing the groups. And the input to this one has to be logarithmized data, sorry. And basically, it's just going to be doing the differential gene expression. So it's going to be comparing the groups of, like the expression of the genes of one niche, for example, tumor 1, against the other two tumors to identify genes that are overexpressed or underexpressed. Like, oh, yeah. So well, running this one, we just compute that, and then we need to run the next one just to get an idea of the genes that we have. So for example, in here, I get MMP1, SP1, NK1, and then IL1R2, etc. And then we see different genes for tumor 2, and then different genes from the border. Like this can help you to further identify like what's going on the different tumor tissues. You can do pathway analysis, enrichment analysis, like it's up to you. So after we identify the top genes, we can just visualize it as a data frame. So we're going to tell it from the rank genes that you identify and the results, just generate a data frame and show the top
This
10.
is a very, I mean, this is a very straightforward analysis that we all do, right, in bioinformatics.
Yes.
So maybe you should show it's fine, but show the results right there. Ah, this is a slide right there. That is too small in my screen. Okay.
Yeah.
Let me see how can I remove this. Okay.
Okay. So now that we identify the genes, we can, for example, select, and let me put it like this, the top 3 genes for each of the niches that we are studying. So here it's showing the genes, and then it's showing also the p-values. So you can check if it's significant or not. So let's start,
Yeah.
and let's put the top
We
3.
don't see that p-values. I don't see that.
These are the p-values. Tumor 1 underscore p, tumor 2 underscore p, and then tumor stroma border underscore p. These are the p-values.
I don't know. We are in the same slide. What is slide? What is the title of your slide?
Can you see my screen? Are you looking at my screen sharing?
Supposedly, downstream analysis is the title of the slide.
Downstream analysis is the title of the slide. And then we just obtain the results in a table.
S3 step 14.
And then if I go to the code, do you see it?
No. No, I don't see it.
Everyone else can look at my code.
Yes, just verify that because I see the same slide and I don't see change. I think nobody's here anyway.
What about now?
Well, now I can, yes.
Okay.
So, I mean, I think the people were not looking at your screen also. So try to go to the results. This is a very standard analysis, right?
Yeah, I already showed them like the microenvironment results.
Yes, the results of this that you are showing now.
And then the results of these, yeah, well, here I was just explaining them that we can get the top genes. So let me just put them in here. So we can visualize them as a dot plot to observe the differences. I can upload the slides to the Dropbox so you can have them. Yes, I'll do it after we finish the tutorial.
So you don't have this in the slides already, Carla?
This
These
part, yes.
results?
These
Okay.
results, yes. But they don't have the slides. They were just, someone was asking on the QA if I can provide them. So we're gonna paste the genes in here. Right now I'm just selecting the top 3, but if you want, you can put more or less genes. Again, it's up to you depending on the analysis that you want to do.
This is enough. This is enough.
Yeah. And then we can finally plot a dot plot where we can see how these 3 genes are highly expressed on the niche 1, the next 3 genes on the niche 2, and the last 3 genes on the tumor stroma border.
Okay.
And this
I
is
think
it.
anybody has a question, please answer in the text in the chat. Anybody has a question? I think nobody's here anymore.
Oh yeah, they're here. They're actually running it.
Well, nobody answers. So get it. What is the next one?
So at the end, we just have the entire pipeline together, so you can just like take a look at it.
Okay. Well, this is the tutorial. Any questions? You have also all the data in the folders. We will put these slides also there. And if you have questions, you can reach my email. Here I'm right now writing in the chat. In fact, I don't have a. Okay, the questions and answers. If there are no more questions, I will say thank you for your attention. This is the tutorial. This is the end of the tutorial.